Hello dear participants, we are in the SWAM course where we will be talking about the introductory animal physiology. In this, first we will talk about the blood. This is lecture of week 1, lecture number 2. We will be talking about the blood and blood as a body fluid. I am Dr. Manish Kumar, Assistant Professor, Department of Veterinary Physiology and Biochemistry, Faculty of Veterinary and Animal Sciences, Institute of Agricultural Sciences, Banaras Hindu University. In this topic, we will be covering about blood, constituents of the blood, plasma and its functions, serum, plasma protein, red blood cells and its composition, shape in the different domestic animal, functions of the blood, Hemopoiesis and Erythropoiesis. Blood is a fluid connective tissue that flow all over the body. The vessels of cardiovascular system is generally around 8 to 10% of the body weight. It consists of cellular element and fluid. Fluid is known as plasma, which is in which the cellular elements are suspended. Cellular elements are erythrocytes, which helps in the transport of oxygen and carbon dioxide. Leukocytes, that is also known as WBC, which mainly perform the defense work in the body. Thrombocytes or platelets, it involves in the blood coagulation. Plasma constitute about 55 to 70% of the blood volume, while the cellular component account roughly around 35 to 45% of the total blood volume. What are the constituents of the blood? On the gradation of the blood color from bright red to the bluish purple are seen depending on the degree of saturation of hemoglobin with the oxygen. On centrifugation of the whole blood containing an anticoagulant, the components are separated according to their relative specific gravity. Cellular component, that is erythrocytes, leukocytes and platelets, occupy the lower portion of the tube. Plasma occupies the top portion and is the liquid component of the blood. Colloids are suspended and other substances are dissolved. On centrifugation, you can see the different elements separately in the tube. Leukocytes are classified into two different groups, that is granulocytes and agranulocytes. Granulocytes means they have the granules in their cytoplasm which can be stained with the different stain and can be visualized under the microscope. Whereas agranulocytes, they do not have the granules in their cytoplasm. If at all it is there, it is a very small and very minute. That's why they are called as agranulocytes. Granulocytes originate from the lymphoid stem cells known as lymphoblast in the lymph tissue. According to their staining reaction, these cells are classified as neutrophils, eosinophils and basophils. Neutrophils incorporate both basic and acidic component into their granules, whereas eosinophil only accept the basic component of the stain. Basophil only accept the acidic components. And agranulocytes are lymphocytes and monocytes. Lymphocytes nucleus is filled completely, very less cytoplasm will be there. In the monocyte, the nucleus is of little bit kidney shape. If you talk about the total number of leukocytes, it is being measured like per microliter of the blood and percentage of the each leukocytes. So, if in this you see that leukocytes, basophil are the least number in the blood. After that, eosinophil comes around 2 to 5%. We will be rarely seeing that basophil when we make the smear and stain it. and observe under the microscope. Monocytes are around 5 to 6%. Lymphocyte, it is 50 to 60% in the pig, and all the ruminants are having the more lymphocyte as compared to that of the neutrophils. Whereas in the dog, cat and horse, you will be seeing more neutrophil than the lymphocytes. In chicken, it is around neutrophil is 25 to 30% and lymphocytes are 55 to 60% and monocyte 10%, eosinophil 3 to 8% and basophil 1 to 4%. Plasma, it constitutes about 55 to 70% of the total blood volume. Plasma is yellow to colorless depending on the quantity, the species of the animal and its diet. Plasma color is golden yellow in the case of dog. Cow and especially in horse, it is usually darker in color. The color chiefly result from the varying concentration of the pigment that is called as bilirubin and to the some extent by the carotene, xanthine and other pigments. Composition of the plasma. In adult animal, plasma contains around 91 to 92% water and 8 to 9% solid. Over 7% of the plasma solid consists of plasma protein alone. Rest 1 to 2% include the non-protein nitrogenous compound. If you see that it has the various component like organic components, it includes carbohydrate, glucose, lactate and pyruvate. Inorganic component, that at micro level, iron, and micro level it is manganese, cobalt, copper, zinc and iodide. Plasma protein includes albumin, globulin and fibrinogen. Non-protein nitrogenous compounds urea, uric acid, creatinine, salt of ammonia. Lipids include neutral fat, cholesterol, phospholipids, lecithin. Others, enzymes, hormone and vitamins. These are the composition of the plasma. Serum. Serum is a watery fluid. Once the blood is oozing out from the blood vessels, blood will clot, and after retraction of the blood, that watery fluid will ooze out from the clot, that is called as serum, and it is devoid of the clotting factor such as fibrinogen, prothrombin and other coagulation factor which are involved in the blood coagulation. They are devoid of this. This is called as serum. pH of the blood is maintained normally around 7.4. The venous blood is slightly towards the acidic side, that is 7.36 because of the increased content of carbon dioxide, whereas arterial blood is slightly more towards the alkaline side. Plasma is more alkaline than the corpuscles. The blood pH range in different domestic animals are like in dog, it is 7.32 to 7.68, in cattle 7.35 to 7.50, in horse 7.35 to 7.43, in fowl 7.56. Plasma protein, there are three major plasma proteins present in the plasma are albumin, which constitute around 55% of the total protein, globulin, it is around 38%, fibrinogen, it is around 7%. The ratio of albumin to the globulin is 1:0.7. Species variation in the ratio of plasma protein, that is albumin to the globulin, is there. In the sheep, goat and dog, the albumin predominates. over the globulin. In horse, pigs, cow and cat, albumin and globulin ratio is equal. In newborn animals, except rodents and primates, the plasma gamma globulin is absent or found in a very minute quantity. What are the functions of plasma protein? It is acting as a source of amino acid for the synthesis of tissue proteins. It provides the colloidal osmotic pressure and helps to regulate the fluid balance. Around 80% of the colloidal osmotic pressure is contributed by albumin alone. Plasma protein also act as a blood buffer and regulate the acid-base balance and maintains the normal pH of 7.4. Fibrinogen and various clotting factors are essential for coagulation of blood. It contributes to the viscosity of the plasma by gamma globulins. Plasma proteins also act as a carrier protein. They are involved in transport of copper, iron, heme, bilirubin, xanthin, cortisol, sex hormone, vitamin A, D, fatty acid, triglyceride, phospholipid, cholesterol and other substances. Red blood cells. Red blood cells are also known as erythrocytes. In the circulating blood of the mammals, red RBCs are non-nucleated, non-motile cell. On combining with the oxygen, they form the oxyhemoglobin, which is a bright red in color. The number of erythrocytes vary greatly among the different species as well as within the species and within the individual of the species. Factors like breed, age, sex, exercise, nutritional status, lactation, pregnancy, excitement, blood volume, stage of estrous cycle, time of the day, environmental temperature, altitude and other climatic factor also influences the number of red blood cells in the individuals. Cell count vary as well as between the arterial and venous blood sample. Origin of the blood cells. In early fetal development, nucleated red blood cells are produced in the yolk sac. During the embryonic development, liver, spleen, lymph node are the source of the RBC production. As fetus grows and later part of the gestation and after birth, erythropoiesis takes place in the bone marrow of the different bones. Marrow of the membranous bone, that is body of the vertebrae, pelvis, ribs and sternum, remains active even in the older age, and in the older age these flat bones are mainly source of RBC production. If you talk about the number of RBC, that in the different domestic animals have the different range of RBC in the and it is being expressed as a million per cubic millimeter. Cattle and pigs have the 6 to 8 million per cubic millimeter, horse around 7 to 12 million per cubic meter, sheep 10 to 13 million per cubic millimeter, dog around 6 to 8 million per cubic millimeter, chicken 2.5 to 3.2 million per cubic millimeter, in pigeon around 3.5 to 4.5 million per cubic millimeter. If you talk about the shape of RBC, it is usually biconcave circular disc with the central pale spot is there. Shape of the RBCs varies in the different species like dog, cow and sheep. It is markedly biconcave. Whereas in the horse and cat, it is a shallow concavity is there. In the goat, it is a very shallow or flat surfaced erythrocyte is there. In the camel and deer, it is elliptical in shape and sickle shaped. Cold-blooded animals and birds have the elliptical and nucleated RBCs. In the mammals, you will not find nucleated RBC, whereas in the birds and cold-blooded animals, you will find the RBC having the nucleus. Biconcavity increases the surface area, thus facilitating the exchange of the oxygen and carbon dioxide carried by the RBC. To facilitate the transport of the carbon dioxide, the RBC contains the enzyme that is called as carbonic anhydrase, which helps in the transport of the carbon dioxide and oxygen. Then coming to the size of the RBCs, the average diameter of the RBCs ranges from the 4.1 to 7.3. Surface area varies around 57 to 67 meter square per kg body weight. Among the domestic animal, it is the lowest size in the goat and the highest in the dog. That is, in the goat around 4.1 micrometer in size, whereas in the dog it is 7.3 micrometer in the size. Composition of RBC. The principal component of erythrocytes is a hemoglobin, which makes up about 1/3 of the erythrocyte content, the remainder being water and stroma. Erythrocytes in adult animal contain 6.2 to 7.2% water and remaining approximately 35% is solid. Hemoglobin constitutes about 95% of the solid. The major solid of the other 5% are protein in the stroma and cell membrane, lipid, vitamins acting as coenzymes, glucose for the energy, enzymes and minerals are there. Sodium is the principal cation in the extracellular fluid and potassium is the principal cation of the erythrocyte of some species. Specific gravity of the blood is the ratio of the weight of the blood to the weight of the same volume of the water at a temperature of 4 degrees centigrade. It depends upon the PCB and hemoglobin and protein content of the plasma. A specific gravity is measured using the instrument that is called as refractometer. In animal, the domestic animal like goat, it is a specific gravity is 1.042, pig 1.045, dog 1.045 to 1.052, cat 1.050, sheep 1.051, cattle 1.052 and horse 1.053. Talking to the lifespan of the RBC, it is different in the different animals. Again, normally about 1% of the red blood cells are replaced daily based on the fact that they live approximately around 100 to 120 days. Erythrocytes are destroyed in a great number daily. The total number of erythrocytes in a 450 kg animal with a blood volume of 8%, a total body weight is around 300 trillion. Considering the average life of erythrocyte being 100 days, the 3 trillion must be destroyed and formed every day. That is about 35 million per second RBC need to be destroyed and formed. Here we can see that lifespan of RBC in the different animals. We can observe that in the dog it is 100 to 130 days, cat 70 to 80 days, pig 51 to 79 days, horse it is 140 to 150 days. This is the, I think they are the highest lifespan, and the all adult ruminants also it is around 125 to 150 days they live. Whereas lambs and calves it is around 50 to 100 days. In chicken and duck it is around only 20 to 30 days, and in ducks 30 to 40 days. Rabbit and rat it is 45 to 50 days. In human it is around 120 days. Short lifespan of the erythrocytes in the birds is due to the high body temperature and rapid metabolic rate. Coming to the functions of the blood, the major functions of the blood is transportation. It involves in the transport of oxygen from the lungs to the tissue and carbon dioxide from the tissue to the lungs. It also involves in the transport of the nutrients from the GI tract to the liver and other cells. Transport hormones from the endocrine glands to the target cells wherever it is required to act upon. It also transports the waste product from the cells of the excretory organs to the from where it can be disposed. Blood also regulates the maintenance of homeostasis. It also regulates the pH, regulates the acid-base balance. It regulates the body temperature and water balance. It regulates the somatic pressure, and blood also involves in the providing the immunity. Like different white blood cells, their major functions is to provide the immunity to the body. Coming to the hemopoiesis in the animal, that all the blood cells, either it is erythrocytes or it is WBC or RBC, platelets, all the blood cells are being formed from the undifferentiated pluripotent stem cells. This is the stem cells from which all the blood cells are being formed. And from undifferentiated pluripotent stem cells, it derived the myeloid stem cells. From this myeloid stem cells, that it divides into the megakaryocytes. Then from that megakaryocytes, it forms the platelets, and that myeloid stem cells form the granulocytes precursors. Granulocytes precursors produces all the granulocyte cells, that is basophils, eosinophils and neutrophils. These are produced from the granulocytes precursors. Myeloid stem cells also produces the erythrocyte precursors, and from this that erythrocytes are being produced. And from the myeloid cytes, it also monocyte precursors are being formed, and that monocyte precursors are again converted into the monocytes. Whereas lymphoid stem cells is derived from the undifferentiated pluripotent stem cells. They are responsible for the formation of lymphocytes in the lymphoid tissue, and it is involved in the formation of lymphocytes, that is B lymphocytes and T lymphocytes. All these lymphoid cells are being formed from the lymphoid stem cells. So if you see this, that myeloid cells are responsible for the formation of the granulocytes, all the granulocytes, platelets, erythrocytes and monocytes. Lymphoid stem cells forms only the lymphocytes, that is T lymphocytes and B lymphocytes. Erythropoiesis, that is formation of or genesis of RBC, the first cells identified as belonging to the red blood cells series is proerythroblast. This is the proerythroblast is the cells which belongs to the red blood cells series. Under the appropriate stimulation large number of these cells are formed from the colony forming unit stem cells. The proerythroblast divides multiple times, eventually forming the many mature red blood cells. The first generation cells are called as basophil erythroblast. This is the first generation cells are called as basophil erythroblast because they stain with the basic dye. The cells has accumulated very little hemoglobin at this stage. In succeeding generation, cells become filled with hemoglobin to a concentration of about 34% and nucleus condensed to a small size. Just before the entry into the circulation, nucleus and endoplasmic reticulum is absorbed and expelled from the erythrocytes. The polyribosome and ribosomes are retained in and might still be apparent for a day or so after their arrival in the circulation, and they are known as reticulocytes. Polyribosomes consist of several ribosomes joined together by the same messenger RNA molecules. During this reticulocytes stage, the cells passes from the bone marrow into the blood capillaries by diapedesis. The remaining basophilic materials in the reticulocytes normally disappears within 1 to 2 days, and the cells are then called as mature erythrocytes. Because of the short life of the reticulocytes, their concentration among all the red cells of the blood is normally slightly less than 1%. During the period of rapid RBC production, reticulocytes number can increase. Reticulocytes are usually present in the blood of animals when the lifespan of erythrocyte is less than 100 days. In adult ruminants, especially horse with the longer RBC lifespan, do not have the reticulocytes in their circulating blood in a healthy condition. So, if lifespan of the RBC is less than 100 days, reticulocytes in the blood can be seen easily, whereas if the lifespan of the RBC is more than 100 days, the chances to see that reticulocytes in the circulation when the animal is healthy is very less. That is the case especially in all the ruminants and horses, where the lifespan of the RBCs are around 125 to 150 days. The nuclei of the avian erythrocytes are not expelled before entry into the circulation, and they persist throughout the lifespan of the erythrocytes. Thank you.